Hello, my name is Brophy. I'm an Associate Professor of Statistics at Trinity College Dublin. I'm going to talk today about the role of multi-species mixtures in intensively managed grassland systems. I'd like to thank the British Grassland Society for the invitation to talk here today. It's a real pleasure to be here this morning. I'd also like to acknowledge my co-authors who are listed on the slide there, and this presentation today is to try to summarise the work that this group has been doing over the last number of years. As a starting point, I'd like to think about a semi-natural grassland system, and a lot of the principles in terms of the biodiversity and ecosystem function relationship have been developed in these types of systems. And what's often found is that as you increase the species diversity, that you have an increasing but saturating relationship with outputs such as the yield. And so when we think of a semi-natural grassland, something like what you can see in the picture there, they can be quite diverse and have quite high species richness. So the question is, do these principles that we've observed in semi-natural grasslands, do they translate into intensively managed systems? So when we think of an intensively managed system, we would think of something more like the next picture there, where we might have a monoculture or a single species zone, and also we might have quite heavy fertilizer input. And so research in this area has started to look at, well, what if we introduce a second species to such systems? Or what if we go further and have multiple species and also maybe increase the functional group diversity? So the question is, can we observe the same BEF principles in these type of systems in more diverse, intensively managed systems? And also, would the multi-species aspect, would it provide some protection against extreme environmental disturbance, such as maybe an extreme drought or an extreme waterlogging event? So I'm going to talk today a little bit about the background in terms of extensively managed systems. We're also going to talk through the last 10 plus years of how this work then has transferred into intensively managed systems and what's going on there and what are the questions that we still have to answer about this. So I'm going to start with the agrodiversity study, which is a study that was carried out in the early noughties. And this study was a European-wide experiment, and there was also one site in Canada. So at each site we had a common experiment. And that common experiment consisted of 2 grasses and 2 legumes. And within the grasses and within the legumes, one species was fast establishing and the other was temporally persistent. So given the wide climatic gradient here, the exact same set of species were not used at each site. But here's an example of one set of species that were used. So we have the 2 grasses, Lolium prenni and Dactylus glomerata, and the 2 legumes, Trifolium repens and Trifolium pratense. So the choice of species was chosen by the individual site, but the functional structures of the 2 grasses and the 2 legumes and the establishment rates within that were consistent across all of the sites. So the experimental design at each site was what we call a simplex design. So what we had monocultures of each of the species, and we also had 4 species mixtures where we varied the proportions of the 4 species. So we had some plots where 1 species was dominant, some plots where 2 species were co-dominant, and then we had the equiproportional mixture. And this simplex design was repeated at 2 densities at each site. So across 31 sites with 30 plots at each site, we had 930 plots in total. And these were measured generally over 3 years at most sites. So I'm going to go through some of the key results from this study. And this is the first one here. So what we have is the total yield over the 3 years on the y-axis, and we have site on the x-axis. So for each individual site, what we're looking at, I'm just going to hone in on one of the sites here. What we have here, these are the mixture averages, and it's averaged across the 2 density levels. And what we have here is the average of all of the monocultures. And here we have the average of the best monoculture. So what you can see is out of the 4 species mixtures, regardless of whether they were co-dominant or equiproportional or dominant by 1 species, we're generally seeing that they were performing better than the average monoculture, but also better than the best performing monoculture. And that was more surprising. So the star oval on the top is indicating where we have transgressive overyielding, where the mixtures were outperforming the best monoculture, and so significant transgressive overyielding. And we saw that at the majority of sites across the 3 years. So that's just focusing in on the yield. But if we want to think about it from a multifunctional perspective, what I've presented here are just the raw data means. So this is on average across all sites and all years. And what we're seeing is positive mixing effects on 4 different responses here. So I'm just going to hone in on the total biomass. So what we have here are the 4 species mixtures where 1 species was dominant. Here we have the equiproportional, all 4 species are equal in equal proportions. And the next ones we have, these are the co-dominant mixtures, where two species are co-dominant. The purple then are the grass monocultures, and the oranges are the legume monocultures. So what we can see is a consistent outperformance in the mixtures, regardless of which of the 4 species mixtures compared to the monocultures. We also see that for the sown biomass. This is where we exclude the weed biomass. And we also see it for the N biomass as well. And here what we're seeing is that the weed biomass is very much suppressed across all of the 4 species mixtures compared to the grass monoculture and compared to the legume monoculture in particular. So what we're seeing is positive mixing effects across multiple functions here: the biomass, the total biomass, the sown biomass, the N biomass, and indeed the weed biomass. And so from a practical perspective, of course, another question is, well, how do these mixtures affect the forage quality? So again, here, I'm just going to show raw data averages. And what we're looking at here is a subset of 16 sites. And what you can see is quite a flat relationship, really, particularly on some of these functions. So what we're seeing here is that the mixtures are not very different to the monocultures. So what that's telling us is, so initially there may have been some concerns that introducing mixtures would reduce the forage quality, but certainly this is showing that on average across all of these many sites, that there is no negative impact of mixing on the forage quality. And that's across multiple forage quality value variables. So the agrodiversity study, in a way, it was really a landmark study. And in fact, it's a real pleasure to talk about the agrodiversity study, because this was really my introduction into research. The week before I started my PhD, I was involved in a training event for many of the researchers from this network. And so it was a fantastic introduction to research for me at the very beginning of my research career. And in fact, I met one of the participants from this agrodiversity network at the last in-person conference that I went to back in the EDF in 2019. And once we hadn't seen each other since the time of this experimental work, and once we got reacquainted with each other, kind of looked at me and said, you've really grown up. And I think it's really true. I think that this network was the very beginning of my research career, but in preparing this presentation, I really kind of reflected on how it has shaped some of the directions that I've gone in my research work. And it really was a landmark study. And so I've just listed some of the publications there that have arisen from the agrodiversity study for further information. So the question is, where did we go from the agrodiversity study? What were the next questions that we really needed to ask? And what I'm going to do to address this over the course of the following 10 years, what did this group do to to advance the findings from the agrodiversity study? And so I'm going to just do a little bit of a whistle-stop tour of a few studies that this group has looked at over the last 10 years. So what were the directions compared to the agrodiversity study? What were the directions that we went in? So first of all, we looked at increasing the functional group diversity. So the agrodiversity study, we had grasses and we had legumes, and now we've looked at the introduction of herbs as an additional functional group. We've also looked at increasing the species diversity. Of course, in the agrodiversity study, we had 4 species. So are there additional benefits to increasing that up to maybe 6 species? We've also looked at how mixtures protect against environmental disturbance, and we focused in particular on drought. We've also looked at how N fertilizer manipulations will affect the relationships. And we've also studied multiple functions and tried to move away from just the focus on the yield. And at the same time, there were many statistical developments going on to analyse the type of data that these studies were generating. I'm going to start off by looking at some of the drought experiments that we have done. And so to do these experiments, what we do is we impose experimental drought shelters on the plots, something like what you can see in the picture up the top here. And you might kind of question, why would we do drought experiments in Ireland? You're having buckets loads of rain in Ireland. And here's an example of a drone shot from one of these studies, and you can see the checkerboard effect there of the diversity gradient. And the question, why are we studying drought in Ireland? But actually, this is the exact same experimental site in 2018, and you can see very clearly that we're in the midst of a severe drought, and you can see just how badly affected the surrounding region there is by that drought. So, in Ireland, we are certainly not immune to the extreme climatic events that are occurring. And, in this experiment, we actually had to irrigate our control plots, which was certainly not expected in Ireland. But when you think about it, it's maybe perhaps not so surprising. So I'm going to look at 2 different studies. The first one was a 2-site study where 4 species were used. And the 2 sites, one was in Ireland and one was in Switzerland. So at each site, we had a simplex design, and this simplex design, we had monocultures of each species, we had 2 species communities, and we also had some 4 species communities where 1 species was dominant, and also the equiproportional community. So the number of replicates across those communities depended on the community. And also then there were rain-fed plots and there were drought treatments where we imposed the experimental drought. So in total, there were 70 plots in each site. So what did this study find? What were the results from this study? So I'm going to focus in first of all on the control side. And what you can see here is what happens to the yield as we increase from 1 species up to 4 species. And we can certainly see an increasing positive relationship there as we move from 1 to 4 species. And what happened then under the drought? We can also see that, of course, first of all, that there was a shift down, as you would expect. There was a decrease in productivity, but we can still see an increasing relationship across the diversity gradient when we focus in on the richness. And just something to highlight here really is if we look at the monoculture in the control for the rain-fed plots, we can see then that, of course, in terms of the comparison between the monoculture and the drought, there is a shift down. But we can see that the 4 species mixtures are they're performing comparably to the monoculture, and here they're actually outperforming the monoculture in the control. So even under the drought conditions, the 4 species mixtures are outperforming the rain-fed monoculture conditions, and that's quite a strong result. So this work then moved on to a new experiment which was looking at 6 species. And this experiment was carried out at 1 site, and again we used a simplex design where we varied the proportions of 6 species this time. And again, the design that's shown here, there were different replicates depending on the community. And what we also had was a split plot design where there was a rain-fed and drought treatments within each main plot. So there were 86 subplots in total in this experiment. So here are some of the results from this experiment, and I'm going to focus in on the yield first of all. So what we have here is the dry matter yield, and you can see here these 2 ternary diagrams. So how do these ternary diagrams work? So for the corners of these ternary diagrams, so here we have grass, here we have legume, and here we have herb. What we have in the corners is where we have just a single functional group. And so in the top part, this is representing what would happen when you have 2 grasses mixed together. And then as you move around the ternary diagram, you're changing the relative proportions of the functional groups, but you're keeping the 6 species in there all the time. So for example, if we take this point here is the center of the ternary diagram, and it's representative of the 6 species equiproportional mixture. And this is what the prediction is here for that community. And under the drought conditions, we can see the equiproportional community then is over here. It's a little bit lower. So what is quite interesting here is that we can see that there is certainly a mixing effect and a positive mixing effect. So as we move towards kind of the center of the ternary diagram, we are seeing increased yields, but it is quite robust, and we're seeing quite a large area is performing very, very well. But we're also seeing that this diversity effect is present under the drought conditions, and there are strong mixing effects under the drought conditions as well. And quite an important comparison here was in this study, we also looked at higher fertilizer level of just monocultures of Lolium perennae. And this is how that monoculture performed at both rain-fed and drought conditions. There was very little difference between the rain-fed and drought conditions for the highly fertilized Lolium perennae. So all plots in this experiment were fertilized at 150 kgs of nitrogen. But these monocultures of Lolium perennae were fertilized at 300, so double that. So what you can see is that multi-species mixture under the rain-fed conditions is quite considerably outperforming the highly fertilized Lolium perennae monoculture. And even under the drought conditions, what we're seeing is that this 6-species mixture is performing comparably and slightly better than the high input Lolium perennae monoculture. So that's quite a strong result. And so also some work that's ongoing on these studies is also looking at the emissions. You will have heard from Saoirse Cummins yesterday, and also from Guy Lagrange, who has presented a poster at the conference. He's looking at some of the other functions. And also there's some work going on in terms of looking at the invasion and looking at the N content and other aspects of N. And so there is some multifunctionality work going on this study at the moment as well. So I'm going to move on to another study now, which looked at multifunctionality, and this was a 4-species study, and these were the 4 species that were used. And there was once again a simplex design used here. And this time there was a manipulation of fertilizer treatment across the simplex design. So there were 3 levels of N fertilizer. We had 50, 150 and 450 kgs of nitrogen per hectare per year. And so there was a total of 78 plots in this experiment, and measurements were taken for 3 years. So there were 10 ecosystem functions evaluated, and this work was just recently published in Suter et al. In Scientific Reports. So what did this study find? So first of all, what we have here is we have on the left-hand side here, you can see the range of functions that were studied. And what is shown here on the x-axis is the percentage benefit for the equiproportional mixture over the average monocultures. And the highlight in green is to indicate the preferred direction, kind of like the a priori preferred direction. Like, for example, we prefer to have positive yield, but we prefer to see less weed biomass. And so what you can see straight away there is that, first of all, the direction of the estimates and the confidence intervals are all in the direction of the preferred direction. They're all in the green for the most part. So what we're really seeing is we're seeing increased multifunctionality with just 4 species in this agronomic system. And there's no reduction in forage quality at the 2 lower N levels, the N50 or the N150. And so generally the high N fertilizer reduced the beneficial mixture effects. So you can see kind of a decrease here, for example, in the beneficial mixture effects as you increase the fertilizer level. So of course, these species were targeted and they were chosen for their complementary traits. And I think that's one of the benefits when it comes to intensively managed systems, is that we can focus in on the species, and we know lots of information about their competitive abilities and about their performance abilities. And so we can pick select targeted combinations of multi-species mixtures. So also alongside this experimental work, there have been many statistical developments going on, in particular the analysis of the simplex design that I mentioned a few times. And what we're really trying to do with that statistical analysis is go beyond the number of species as the sole explanatory variable for what's happening in multifunctionality. And so to look at things like species-specific effects and their interactions. And also, how do we design multi-species mixtures? So there's some work going on in terms of optimization of multifunctionality. And so also, if anybody has some data that they would like to analyse themselves, we have a new OR package called the DI models package, and that's suitable for analysing this type of data from a simplex design. So that's the work that's been going on since the agrodiversity study. It's really just a snapshot of some of the work that's been going on by our group. And of course, there are plenty of other studies out there outside of our group. So it's really just to kind of touch on some of the studies that have been going on. So what are the remaining questions? What are the next steps for this field? Where do we need to go next? So I'd like to first of all introduce a new global network of experiments called LegacyNet. And this is a network that has been established by myself and John Finn and Karsten Mallet in the last couple of years. And really this network is looking at what is the role of multi-species mixtures, multi-species grassland lays, if you like, within crop rotations. And so far we have 24 sites established. You can see the locations of our site there around the world. We also have 14 more sites included there that are committed to establishing this year. And of course, more sites are welcome. Should you know of anybody who is interested in joining our network, we're still welcoming new members until the end of this year. And our website address is there. So just a little bit more about this experimental network. We have 2 phases. The first is a grassland lay phase, where we will have a simplex design and a species diversity gradient across 6 species. So we will have, for example, monocultures of each species. We will have binary communities of each species. We will have 3 species communities, 4 species, and the equiproportional community as well. So what we will do is we will establish the grassland lay phase first of all, and the multi-species mixtures will grow for at least 18 months. And what will happen then is on the individual plots, the grassland stage will be taken up and a follow-on crop will be sown. So the follow-on crop will be sown uniformly across the plots, but of course we will know what the previous grassland lay mixture or monoculture was before the crop was sown. And so The questions that we're going to ask, first of all, will be related to the grassland lay phase and how will species and functional group diversity, how will it affect the outcomes on the grassland lay stage? And of course, these results, because we're going to have this global experiment, we're going to be able to address these questions on a really large scale and across a spatial scale. And that's one of the things, I mean, single site experiments are great, but how can you replicate the findings across multiple sites? And that's one of the real strengths of networks such as the agrodiversity study, but also such as LegacyNet, where we can test these effects and test how robust they are across a very wide spatial gradient. So the multi-site aspect here is key. And also we will focus in on multifunctionality as well. Of course, we will look at yield outputs, but we will also look at lots of other outputs as well. And of course, then when it comes to the follow-on crop phase, one of the main questions that we will want to ask is, are there legacy effects of the species diversity gradient? Do we see different effects of, for example, the monocultures compared to the 6-species mixture on the follow-on crop? Are there legacy effects there? And do they provide a boost, for example, do they provide a boost to the follow-on crop? And I think, yeah, there's lots and lots of potential with this site and with this experiment and the types of questions that we can address with it. So what's next for intensively managed grasslands? In terms of further experimentation, we've kind of seen, you know, the landmark study of the agrodiversity study and where we've gone from there. Of course, we are interested in the role of multi-species mixtures in crop rotations, and LegacyNet will address, the LegacyNet network will address that. There's also further work to be done in terms of other environmental disturbances. The work I showed here quite focused in strongly on the drought impact, but of course there are many other biotic or abiotic environmental disturbances that we could examine as well, and that are being examined already. Of course, mechanisms, like what are the mechanisms by which some of the processes are happening? What are the practical implications, and how, for example, do some of the experimental results that we've observed already, how do they translate into grazed systems? And there is some work going on in this space already, and there's plenty more to do. For me personally, I think multifunctionality is key. We need to move away from focusing on purely the yield outputs, and I'll start to think about the bigger system. You know, if you have a highly fertilized monoculture, it's a leaky system. Where are we losing? We're losing emissions, we're losing nitrate leaching, we're losing all sorts of, we're losing from the system. Whereas when we have multi-species mixtures and we think about it from a multifunctionality perspective, we can really look at it from a much more holistic viewpoint. So, you know, from a policy and a sustainable agriculture perspective, of course, we all know that fertilizer inputs have to reduce. And multi-species, what this work is showing is that multi-species mixtures do offer great potential when it comes to sustainable agriculture. So what are the take-home messages? So first of all, the agrodiversity study, we showed that 4-species mixtures improved yield, N biomass, weed suppression, and also that there wasn't a reduction in forage quality when we moved towards the 4-species mixtures. In terms of the other studies that we've looked at, we've shown that increasing species and functional group diversity promotes multifunctionality and also provides protection against extreme environmental disturbance. And in terms of the future, I think, you know, it's clear that multi-species mixture do offer excellent potential for improving the sustainability of agricultural grasslands. So just in terms of leaving you with an image, you know, to think about moving from this kind of landscape that's dominated by monocultures to something that looks like this in the context of intensively managed systems. I think this is something that is achievable. And I think just to kind of summarize it, you know, species and functional group diversity, they promote multifunctionality and can provide protection against extreme environmental disturbance. So I'd just like to thank you very much for your attention, and I'd also like to thank the BGS for the invitation to talk, and I look forward to answering any questions that you may have.